if we were in the conference room, we can see the people, who people is there. So
yes,
but this is the way the world it is now. So
yes.
buenas tardes, bienvenidos nuestra audiencia, tanto de YouTube como por vía Zoom. Vamos a esperar 1 o 2 minutitos para iniciar, en lo que se conectan algunas otras personas. So you are Italian, and so you must understand, like Mexicans, that if it's not 12.
Oh, yes. Oh, yes.
Okay.
Yes, yes. Actually, in Italy, we always have about 15 minutes. I mean, most cases, right? They're called the academic 15 minutes.
Yes.
Meaning in academia, you have 15 minutes usually in which you wait for people to come in.
Yeah, I know.
Those were the old days.
Yes. So now we just wait 1 or 2 minutes. And so how is your laboratory? Are they working?
Yes, yes.
Yeah?
We work pretty much throughout. Of course, at the beginning, not so. I mean, it was slowed down, and we had a lot of rules. But now, pretty much is back almost to normal. Not exactly to normal. I would say about 70%.
Mm-hmm.
So pretty good, pretty good.
Oh, good.
Yeah.
So is almost everybody vaccinated and
Yes, yes. Everybody in the lab is vaccinated.
uh-huh. And do you wear mask outside of the
Yes.
Wow, a lot of people want to hear you. It's 70 participants and it's 12:05. So I want to give the welcome to all the the YouTube persons that are hearing this conference, also to all the persons in the IBNN, and of course, I want to thank you, Dr. Cristina Alberini, for being here. It is an honor for me to introduce her because she has been very kind, as she's going to give a virtual talk in our institute, which is maybe more difficult than in a presential way. I want to tell you that Cristina Alberini received her PhD in immunological science from the University of Genoa, Italy, and then trained in neurobiology as a postdoctoral fellow at Columbia University. And from 1997 to 2000, she was an assistant professor in the Department of Neuroscience at Brown University, and then associate and full professor in Mount Sinai School of Medicine in New York from 2001 to 2011. In 2011, she joined the Center for Neural Science at New York University, where she's currently a professor in neuroscience. She trained as a psychoanalyst and NPAP and became licensed in the state of New York in 2012. This is a very interesting point, I think, from her. We were talking about that because being a psychoanalyst, she can test ideas from that theory in a scientific way. Professor Alberini research focuses on understanding the molecular and brain plasticity mechanisms underlying long-term memory formation and retrieval. Her studies target different ages of the lifespan. The results of her studies provide information for developing potential therapeutics against cognitive impairments and psychopathologies. Professor Alberini received the Hirschwale Career Scientist Award, NARSAT Independent Investigator Award, Golgi Medal, Athena Award, Merit Award, and in 2018, the Jacob K. Javits NYU Award. She has been published more than 90 articles in the field of learning and memory. And actually, she's now the editor of a very well-known journal, which is Hippocampus. So as I say, it's an honor for me to have you here. Thank you so much, Christina. And I let you with the audience, and thank you for the audience to be here.
Thank you. Thank you so much, Gina. Many, many thanks for this wonderful invitation. It's my honor and a privilege to be here. And I'm testing, is the audio good? Can you hear me well? Just making sure. Let me know if there is anything I can do to improve. Maybe I can come closer.
Yeah.
So many thanks. It's so wonderful for me to be here and have this opportunity to be in this audience. Of course, we would love to be in person together, exchanging information, but hopefully this will happen soon for a next time we can meet. I'm going to start by sharing the screen so that we can get into my presentation. As Gina mentioned, my lab is looking at the biology of learning and memory, and today we're gonna discuss one of the questions we have ongoing in the lab, which is the identification of mechanisms underlying learning and memory during infancy. This is a very special time for the brain to start functioning in the learning and memory functions, and very little is known about the biological basis of normal learning at this age. In general, we study this mechanism throughout different ages, not only in infancy. And just to give you a little bit of an introduction to the type of memories we are studying and the type of questions we are studying, I'm going to remind you or tell you, if you're not in the learning and memory field in particular, that when we talk about memory, we actually refer to different types of memories, because we do have different types of memories that we can form. One way to distinguish the type of memories is between short-term and long-term, based on the duration of memory. So we have memories that last for seconds or minutes, and these are the short-term memories. And we have long-term memories that last for days, weeks, years, even a lifetime. And this is something that, an interest that we share in common with Gina. In the long-term memories group, we then have different types of long-term memories. A major distinction is between explicit and implicit memories. The explicit memories are the ones that requires conscious recollections, and the implicit memories are recalled in an unconscious manner. In these two groups, we have different types of memories. For example, in the explicit types, we have episodic and semantic type of memories, and the implicit types, we have different types. Two examples are priming and procedural memories. So the explicit type of memories, and particularly episodic and semantic, are the memories of things we learn in terms of episodic experiences of our life and general knowledge. And the implicit type of memories are included procedural memories, which is how to do things. In the lab, we are interested in mechanism of long-term memories, and we use episodic memories as models to study the biology underlying long-term memories. The questions we have in the lab are, throughout the years of our studies, a number. We are interested in identifying the biological mechanisms underlying memory consolidation. This is a process required to form long-term memories, to store long-term memories. Memory consolidation is the temporal window during which the labile representation that is formed initially in an experience becomes stable, becomes stabilized, and requires a certain number of mechanisms over a temporal window. We want to know what type of molecular mechanisms occur in this temporal window of stabilization of memory that allows the memory to become long-term and stable. We want to know where they happen in the brain, how they change over time, which brain regions are involved in these consolidation mechanisms, and which of these mechanisms are critical in different types of brain cells. One bias that we still have in neuroscience is to think about the brain as an assembly of neurons. Of course, we know that that's not the case. We have different types of cells in the brain. We have glia, and we have vasculature cells in addition to neurons. So we have to think about the contribution of different cell types to these mechanisms. We are also interested in the phenomenon or the process called reconsolidation. Reconsolidation is the process of stabilization of a memory that becomes labile after retrieval. So if consolidation is the process that stabilizes a new memory, reconsolidation is the process that restabilizes a memory after it is retrieved. It has been shown that memories can become labile again for a certain temporal window after retrieval, not in all conditions, but in some. And during this period of fragility, memories can be enhanced or decreased, because if we know the mechanisms of stabilization, we can actually change the strength of the memories after retrieval. And we want to understand what type of mechanisms underlie reconsolidation. We also are interested in mechanism of memory strengthening and persistence. These are coming from the consolidation and reconsolidation studies. And the mechanism of memory strengthening and persistence are very important if we want to apply them to diseases that are based on memory loss. And those are many, and also in many cases, devastating diseases like Alzheimer's disease or frontotemporal dementia, or other types of neurodegenerative diseases that lead to memory loss, or even loss of memories during aging. We are particularly interested in different cell types. We have carried out some studies looking at the cooperation between glia cells like astrocytes and neurons in memory consolidation. we are now looking at oligodendrocyte contribution as well as cells of the vasculature. And finally, we are interested in mechanisms of long-term memory in early development, which is the subject of today's lecture. So we're going to talk about this. Learning and memory in early development are very important. As you can imagine, this is when we learn about the world. We are born not knowing where we are and what we do in our life. And we learn a tremendous amount of things and experiences. And understanding the biology underlying these very early memories to me is very fascinating. I became fascinated with this question after I trained as a psychoanalyst, which of course led me to reflecting and thinking about the importance of the upbringing, of the developmental phases, and all the experiences each of us have during our early days. We know from psychoanalytic work, and Freud was, of course, very important in shaping psychoanalysis, in founding and shaping psychoanalysis. We know from psychoanalytic work that clinically we see an important contribution of the experiences of the earliest years of life. Freud says they leave persistent traces in the depth of our mind. And of course, if we talk about clinical material, we can discuss many of the implications of this of this concept of what has been found in clinical cases in terms of how the history shape the way individuals behave, all of us behave. But there is an interesting phenomenon, which is infantile amnesia. Infantile amnesia is the phenomenon by which in adulthood, we don't have recollection of the memories of the first few years of life. So if I ask you, what do you remember of your first 3 years of life? And we are talking here about episodic experiences. So the experiences of facts and people and things and interactions, all of those that we can consciously recollect. What do you remember? And in the vast majority, we do not remember any experience. Occasionally, there is someone says, yes, I do, I have a memory of this. It turns out that it's unclear whether that memory was established later by family members or others telling the individual about these memories. So infantile amnesia is the loss of recollection of memories of the first few years of life. Generally, we talk about 3 years, the first 3 years of life. So there is a paradox. If the early memories shape the behavior throughout life and they leave these persistent traces in the mind of each of us, how is it that we have this persistent effect throughout life if we lose those memories, if the memories are gone, if we forget them? So how can they influence behavior throughout life? So I became fascinated by this number of questions, and to study the biology underlying learning and memory of early life experiences, I have chosen, like for what we do in the adult animals, to focus on hippocampus dependent type of memories. And these are also known as medial temporal lobe dependent memories. These are the memories that I already explained, the explicit types that in humans are called declarative autobiographical memories. So episodic memories and explicit memories in general. These memories require the medial temporal lobe. One brain region that has been highly studied is the hippocampus. We know that the hippocampus is required for this type of memories. If the hippocampus is injured or its activity is blocked in some way, the mechanisms of the hippocampus are blocked, these type of memories cannot form consolidation of these memories is impaired. And these memories can be reproduced faithfully in rodents, for example, because the rodents, like humans, have episodic memories. Episodic memories are memories of that experience in that context with those items, with those conspecifics. So those are the memories of who, conspecifics, people in the case of humans, what, objects, piano, chairs, lights, where, the place, and when, the time that binds all these elements together in an episode. Rodents can process all this information of who, what, where, and when. In other words, they do have episodic memories. So we can use rodents to study the biology of episodic memories in early development. One task we use often is called inhibitory avoidance or passive avoidance. This is a task that I learned from Jim McGaugh, who used this task for many of his studies. It's a simple episodic aversive memory in which the animal is placed into a shuttle box in a compartment with a light, the lid compartment. There is a door separating this lid compartment from a dark side, dark compartment. So after a few seconds that the animal is placed into the lid compartment, the door opens, allowing the animal to go to the dark side of the chamber. And here the animal gets a food shock. We can vary the intensity of the food shock. I can tell you that even with a mild food shock, when we test the memories, and this can be hours, days, weeks later, if they are normal in their memory functions, they develop a long-term memory for this dark context that was associated with the shock, and what they express is a latency to enter into the shock compartment. And this is a typical response we find. We measure the latency in seconds to enter into the shock compartment. On the left of this graph, you see acquisition, which is the time that the animal take to enter into the shock compartment at training before they have received the shock. So they go readily into the shock compartment because they don't know what they find there. But after receiving the shock at testing, in this case, we tested the memory 2 days after training, you see that they develop a very strong, very robust latency. In this case, our cutoff time was 540 seconds, 9 minutes, after which the animal is taken back to the home cage. And you see that there is a very strong memory, very strong latency reflecting presumably the memory. And most of the animal in this case did not enter for the 9 minutes time into the shock compartment. So it's an episodic aversive event that we use. And we adapted this to studying episodic memories in infancy. As I mentioned, infantile amnesia is this inability to recollect episodes of the first few years of life in humans. This happens also in animals. And what happens at the basis of this infantile amnesia is a very rapid forgetting. Infants in humans as well as in rodents learn, and they have memories, but they rapidly forget them. So if we look at the graph on the right, this has been done with rats, and it's looking at the forgetting curve of rats trained in inhibitory avoidance, the task I just showed you. These experiments were done in the 60s by Campbell and Campbell. So they trained groups of rats, and then they started testing at different time points after training, and they measure retention in percent in this case. So that latency is transformed in percent of retention. And what they found is that if they train rats at postnatal day 18 or 23 or 38 or 54 and 100, which correspond respectively postnatal day 18, around 2 to 3 years of age in humans. 23, this is about 7 to 8 years of age. 38 is around adolescence, peri-adolescence. And then 54, a bit older after, and P100 is young adults. So if we train rats at different ages and we look at the retention curves, you see that the rats trained in infancy, a P18, or the juveniles, a P23, they decrease the memory over time. They forget significantly faster compared to 38, 54, and 100. So there is a rapid forgetting, and the most rapid forgetting is in infancy. So this rapid forgetting is well documented in humans as well as in non-human animal lab models. And to try to get to understand what's going on in these early memories, and particularly to give an explanation that will provide some hypotheses to be tested for the rapid forgetting associated to these memories that are lost and relate to infantile amnesia. A number of hypotheses has been put forward initially. The first one to give an explanation for infantile amnesia was Freud. It was not the first one to describe infantile amnesia, but he gave the first explanation. And he says that this is due to repression of infantile memories that have disturbing content of sexual nature. So repression is causing the rapid forgetting. Later, cognitive scientists gave a different explanation and said that it's the lack of language skills and sense of self that at this age occurs that provides this rapid forgetting. So the lack of language skills and sense of self cause these memories to be forgotten and perhaps not consolidated. But rapid forgetting exists in non-human animals, in the lab animals, in rodents. So we need a neurobiological explanation. And a couple of hypotheses, major hypotheses, have been proposed. One is that the hippocampus is too immature and therefore incapable to process episodic information. In other words, the hippocampus is not participating in processing these memories, it's offline. And is incapable of consolidating the memories. And this has been called developmental hypothesis because it says that the development of the hippocampus is not sufficient yet. The other groups of hypotheses are named under retrieval hypothesis and propose that the infantile memories are stored, but that retrieval is impaired. We started to ask a number of questions, and during our work, an interesting first paper came from Paul Franklin and Sheena Jocelyn's group on infantile amnesia, suggesting that neurogenesis, the high rate of neurogenesis during this early age, is interfering with the ability to store memories. In other words, is interfering with the consolidation and storage mechanism, and that's why we have rapid forgetting. But we asked a number of questions in the lab, and we came to slightly different conclusion, although our studies and the studies from Paul and Sheena can actually lead to a complementary understanding of this phenomenon we can discuss later. So we asked these following questions. Are the forgotten memories gone? According to the neurogenesis hypothesis, they should be gone because neurogenesis is interfering with the ability to store these memories. Or is the system unable to express, recall the memories? Which memory system is involved in processing these infantile memories, and what are the type of biological mechanisms recruited if any of these regions is important to form these memories that are apparently forgotten? So the first question is, are they forgotten? Are they gone from the system? Or is there a problem in the system, an inability of the system to express them, but they are stored? So we did very simple experiments. We used inhibitory avoidance at 3 different ages in rats. Postnatal day 17, which I already told you is in the infancy, around 2 to 3 years of age. Postnatal day 24, these are juveniles, around 8 to 10 years of age in humans, in terms of maturation and abilities to function. And adults, young adults, postnatal day 80 to 100. I think in this case, it was around 100. And we measured after training in inhibitory avoidance, immediately after the retention, 30 minutes later, 1 day later, and 7 days later of all these groups. And what we found that immediately after learning, they are all comparable. They have memory, very strong latency, which, however, at 30 minutes differ very significantly. While juveniles and adult rats continue to have a strong memory expression at 30 minutes after training, the rats trained at postnatal day 17, they have almost no memory already at 30 minutes. So they have a very rapid forgetting. This was a single exposure to inhibitory avoidance, a single training trial. At 1 day and 7 days, they continue to have the same. The rats trained at postnatal day 17, they have no memory at 1 day and 7 days. No memory meaning no difference compared to acquisition times. Whereas the juveniles and adult, they have very long-lasting and persistent memories. So clearly the infantile training and memories are very different than memories formed later on. So we focused on these memories and asked whether these memories are truly gone or if there is a problem in the expression. So to try to test whether we could get this expression back, we gave a number of re-experiencing. We started by testing them and exposing them to the inhibitory avoidance box 1 time, 2 times, multiple times. No matter what we did, by testing them once or many, many times, we could never get the memory back. Testing does not get any memory back. We then decided to expose these rats to the shock in a different context. And this approach usually is used in extinction to see whether we can have reinstatement. We did that 1 time, multiple times, shock exposure in a different context. We never got the memory back. But when we mixed these 2 experiences, so first we tested them, and then 2 days later, we gave a reminder shock in a different context. When we tested a day later, these rats, we found that they have a very strong latency comparable to immediately after. Here it is. And when we tested them again, the memory was persistent. So an exposure to the context that was used during training, and 2 days later of the food shock in a different context, so there is no associative memories or associative learning here, returns the original memory to expression. And I'm saying the original memory because we did many control experiments here. If you give a food shock without context at training, at postnatal day 17, you have no memory, you have no recoveries of any inhibitory avoidance response. So you need an associative learning events at training in order to see the recovery of memory after testing and reminder later on. We also tested this memory in a different context, in context B, to see whether there is generalization, and they had no generalization. So this recovered memory is the memory of the context in which they were trained, not to any other context. You can find all these controls and other experiments in the publication. So we concluded that memories, although forgotten, they are only apparently forgotten, or what we can say they are forgotten but not gone from the system. They are stored long-term in a latent form, and we can get them back if we give them certain types of behavioral re-experiences or reminder. So we're going to use this test plus reminder food shock as a reinstatement protocol to test a number of questions now. And so the first question related to mechanisms was, well, is the hippocampus involved? Since the literature suggests that the hippocampus is not involved, it's offline, we can actually now test it. So we're going to inactivate the hippocampus, give this reinstatement protocol and see whether the memory is not there or is there. Or we can do that even for the expression of memory at the reminders protocol itself. So we trained animals at postnatal day 17, injected them in the dorsal hippocampi bilaterally with vehicle or moshimol to block the activity of the hippocampus, and then gave 7 days after training, testing and then reminder shock 2 days later, so the reinstatement protocol, and then tested again a day later. And what we found is that the animals that had the dorsal hippocampi inactivated by moshimol had almost no memory. So memory is impaired by inactivating the hippocampus before training. This experiment told us that the hippocampus is very much involved. The opposite of what the literature was suggesting. It's required to form these latent memories that later on we can get back with reminders. So this is kind of in opposition with the idea that there is a problem with consolidation and therefore the loss of memory. We then tested whether moshimol injection before testing in the hippocampus affects the memory retention, the memory expression. And we found that not to be the case. So the hippocampus is involved in consolidating, storing the memories from the acquisition, from the learning event, but is not involved in the expression when we give the reminder. Probably another region of the brain is involved, and we have evidence that that is probably the medial prefrontal cortex. We also gave moshimol before the reminder shock, and also in this case, there is no effect. So the hippocampus is required for consolidating and storing these memories. So what are the mechanisms that in the hippocampus allow for the storage of these latent memories? We use the semi-unbiased biochemical approaches, semi because we have decided to test a number of synaptic proteins or transcription factors or receptors or metabolism markers or signal transduction pathways that had been identified as important for plasticity in the brain or learning and memory at other ages. And when we did this large survey, we found a number of interesting outcomes. The first one was that learning at postnatal day 17 leads to an increase in the levels in the hippocampi of GLUN2A and GLUN2B. These are the NMDA receptor subunits, A and B, which are important for the functioning of the receptor and the changes of the function of the receptor over development. And when we look at the ratio GLUN2A versus 2B, we found that there is a significant increase in this ratio. This switch in majority of 2B versus 2A, the switch in the increase of the 2A subunit over development was very well known from studies in the hippocampus, plasticity, LTP, LTD, as well as in the visual cortex in sensory systems. And this is a typical change that is associated with critical periods. We're going to get into critical periods definition in a minute. We also look at the BDNF level and the receptor for BDNF, track B, and the phosphorylation of it, and found that whereas BDNF levels do not significantly change with training over time, and over time it means over 2 days after training, 30 minutes, 9 hours, 24 hours, or 48 hours, the level of activation of the receptors significantly increased from 30 minutes up to 24 hours. At 48 hours after training starts to go down to baseline. At 7 days after training is not in this picture. All these changes are back to baseline. This is not exactly what happened at postnatal day 24, which is in the juvenile age. And I just want to remind you that what we found is that the juvenile rats have a long-lasting memory expression like adults. We don't see the switch in the GLUN to A to B. We do see a significant increase in BDNF and a significant increase in phosphotrack B, but with a different profile. As you can see here, is ramping up, peaking at 24 hours as postnatal day 17, and here is peaking at 30 minutes and then going down. So the biological changes we see at postnatal day 24 are significantly different, and particularly the switch of GLUN to A to B over to B does not occur. So this led us to think about mechanisms of critical periods. And we also found, looking at developmental ages, if you compare the levels of phosphotrack B, BDNF, GLUN to A and 2B, at postnatal day 17, 24 in adults, this is non-trained animals, you see that the phosphotrack B decreases, BDNF increases significantly, no change in the 2A at these 3 ages, but a significant decrease in the levels of GLUN to B, and therefore the ratio increases over development. This, again, we just reproduced what in terms of GLUN to A to B was known already over development. We also collaborated with groups looking at the electrophysiological measurements. This is just confirming that there is a switch, functional switch, in the levels of 2B versus 2A using nifemprodil, and looking at the APSC of Schaffer collateral to CA1 synapses from postnatal day 17 and 24, trained or untrained, and found that there is a more significant nifemprodil effect at early ages as at postnatal day 17 with training, consistent with the conclusion that there is a training-induced conversion of a subset of the NMDA receptor from GLUN to B to 2A containing receptors in the infant animals. As I mentioned, this switch occurs over development. It has been observed also in LTP studies and in at early age slices studied with plasticity. And we know that this is important for changing the way that the NMDA receptor works over development. And what has been done also before is that showing that BDNF is regulating this switch. It's a master regulator of synapse maturation and plasticity, including the switch. And these studies have been done particularly on sensory systems and particularly with vision, with the visual system. So all these mechanisms led us to start hypothesizing and test the hypothesis that the hippocampus is undergoing a critical period. What is a critical period? Critical periods are temporal windows during development that shape the functions of the brain. We have critical periods for sensory functions, vision, auditory functions. And these have been studied and shown by many authors. Imprinting is another one. Language is another one. All these functions have been shown to undergo critical period, meaning there is a temporal window during development in which the function has to mature for proper functioning throughout life. If this does not occur in this developmental temporal window, the function will be forever compromised. So I'm going to give you a quick example. You hear me speaking in English with an Italian accent. This is because I learned my English after my critical period for language was closed. So of course the brain is plastic. We can learn new things. We can function, but we're not functioning in the same way as when the function is learned and developed during the critical period. Should I have been exposed to English during my critical period for language, I will speak a perfect English with a perfect accent. And in fact, I do speak, for example, a dialect from Italy. I can go from dialect to Italian because I learned that during my critical period. So, and this is for people who learn multiple languages. That's the case. It's an example. Vision is another one that has been studied a lot. Yeah? If you have a visual problem during the critical period of vision, the vision will be forever compromised if it is not corrected during that temporal window. So what we are saying here, based on our findings and data, is that hippocampal dependent learning and memory undergoes a critical period, like language, like sensory function. So this has a lot of implications if you start thinking about that, right? And we're going to get into the implication in a minute. A number of other mechanisms have been studied in the visual system as mechanisms typical of critical periods. So we went on and tested them. One is BDNF, which shifts the critical period, and the other is inhibition, parvalbumin cells, inhibition through GABA. And the changes in inhibition that can drive the line of opening or closing the critical periods. These are some examples of the papers that had been published in this domain. I don't want to get into the details because I have many other things I want to share with you, but we tested some of these mechanisms. For example, here I show BDNF, which in sensory system, BDNF has been shown to accelerate the maturation during critical period and close critical period much faster, and also be required for critical period mechanisms. So when we injected in the dorsal hippocampi inhibitors of BDNF before training our animals in inhibitory avoidance, and then gave the reminder shock after testing, the reinstatement protocol, we found that disrupted memory. In other words, BDNF is required for these memories, these infant latent memories. And this disruption of BDNF also disrupted the switch of the GLU-N to A to B ratio. Based on the literature, we also tested whether mGluR5, which is known to be upstream of the switch GLU-N to A to B in brain plasticity, was implicated in the hippocampus for our latent memory formation, and turned out that is the case. So when we block mGluR5 using a selective allosteric antagonist, which is this MTEP, in the hippocampi before training, we found that memory cannot be reinstated significantly. There is a significant impairment in memory reinstatement indicating that mGluR5, like BDNF, is required for the storage of this memory and the expression of this memory. And again, the switch was impaired. Conversely, if we give BDNF, now we inject BDNF itself, or we activate with DHPG, the mGluR15 receptors, we found that this activation promotes the expression of memory. As I said, memories are not expressed unless we give the reminder plus the shock, the reinstatement protocol. But in this case, if we inject BDNF after training or the HPG, 1 day later, we see that the animals that received the injection of these compounds had significant memory, and memory keep going up at 7 days at test 2. And these memories are specific for that context. If we test them in a different context, there is no memory. So this is not a memory that is generalizable or generalized to other contexts. And the same is the ratio. We also promoted the ratio of the switch. So in other words, the same mechanism of critical period are functioning in the hippocampus. So we concluded that an mGluR5 and BDNF mediated NMDA receptor switch is required for the formation of latent infantile memories. These mechanisms are typical of critical periods. We also found that similar effects as well as mechanisms, which I'm not gonna list, sorry, are found also in non-aversive tasks like novel object location. So it's not just typical of aversive memories. So then we continued and we looked at other mechanisms and found that learning at this early age, postnatal day 17 here, compared to later, postnatal day 24, shows more differences in the biology. For example, immediate early genes, they are induced slowly but significantly over 2 days, instead of having a rapid peak of induction and return to baseline, which is the typical response of immediate early genes. Immediate early genes that we look at are ZIF268, ARC, and CFOS. So a different way of regulating immediate early genes occur in the hippocampi of this animal. And we continue to look at synaptic proteins and found that, again, there is a very slow increase in synaptic proteins involving plasticity, like synaptophysing or PSD95, compared to no change with training at postnatal day 24. And these changes are required for the memory, because if we knock them down by antisense injections around the time of training, the memory is no longer there. So in other words, from these experiments, we found that learning in infancy leads to long-term memory formation and storage. These memories remain latent, can be reinstated by reminders even later in life. The dorsal hippocampus is very much involved. It is required to form these memory traces. Training in infancy, but not later, leads to a significant increase and switch in the GLUN to A to B ratio, as well as requires BDNF and mGluR5, which bidirectionally can regulate memory formation and maturation. And learning in infancy, but not later, also results in other differential biological mechanism, particularly biological mechanism of synapse maturation, formation and maturation. And these changes are required for the storage of these memories. So we proposed a new explanation for the rapid forgetting of infantile memories. And that is that this rapid forgetting is due to the fact that the system works in a different way with a different biology and uses critical period mechanisms during this time. And that these mechanisms are there to mature the function, very much like in the sensory systems. So the implications here are many. Compromising the healthy development of this critical period would lead to severe impairments in learning and memory functions, like for the vision, the critical period would do. So we need to identify this mechanism because this has also importance for clinical implications and repercussions. So this is graphically what I said. Of course, I expanded on the hippocampus critical period here. We think that like many other functions, somatosensory, amygdala-dependent learning, and later high cognitive functions, there is a critical period for hippocampal functioning. And it is the maturation of this function that will allow further development and expression of critical period of higher or more complex functions. Okay, how am I doing with time? Because I see my watch, I'm getting close to the end.
We're okay, you're close, but it's okay.
Okay, I have one more thing I want to share with you with a couple of slides. And that is the next question we addressed. So if episodic experiences biologically mature the hippocampus, and this maturation is required for the storage of this memory, these latent memories, the next question is, does this experience-dependent maturation develop this hippocampus system, the hippocampus-dependent system as a whole, in general, for all hippocampal functions? Or is the maturation of the hippocampus-dependent system experience selective, experience specific? In other words, is it driven specifically by the type of experiences that we encounter? Or is it a general maturation for the hippocampal dependent types of functions? So we designed an experiment to address this question. And we know from the data I already showed you that training at postnatal day 17, if we test the memories, so these infant memories after 1 day or 6 days, there is no memory expression. We need to provide them with the reinstatement protocol to see them back. Otherwise, they don't express. There is no memories, they are latent. But we also know from our data that there is a significant development of the biology of the hippocampus after training, which occurs in the first 24 hours. I showed you all those markers ramping up during the first 24 hours, even 48. So if there is a maturation during the first 24 hours, perhaps capturing this maturation is the way to test whether we can mature the function. And we did exactly that. So we gave a second trial, learning trial, a day later, postnatal day 18, after the first one was given at postnatal day 17, and measured the memory. And what we found that, in fact, after this second trial, when we test the memory is expressed now. It's no longer latent. It's expressed. It's mature in terms of the behavior. We tested these rats also in another context to see whether this maturation captures an inhibitory avoidance representation in a different context. And we found that there was no latency, no memory for that. So the maturation of the second learning trial is context specific. Now we gave the first trial at postnatal day 17, the second at postnatal day 18 in a context B. These are two different contexts now. And we tested them in A and B. What we found in this case, that the rats have memory for both. In other words, the maturation of the similar experiences, but two different contexts, leads to the maturation of the memory for both. So there is a not generalization, because as you can see here, they didn't generalize, but there is a transfer here of the maturation to a similar type of memory, but in a different context, if they had that experience. Now we repeated the same with novel object location to see whether non-aversive memories do the same. In this case, we test them earlier because it is a shorter term memory. 2 hours after training at postnatal day 17, they have no memories. We know that. But if a day later we give a second trial and then we test them 2 hours later, we found that now they have memory for this configuration and not for another one. Now, when we give 2 different configurations, postnatal day 17, one configuration of the object, at 18, a different one, and then test, now we see that they have memory for the second configuration as well as for the other one. So in other words, they behave the same as the inhibitory avoidance. If we give a similar experience repeated, although the configuration is different, they develop competence for both of these experiences. Now the question was, if we give 2 different types of hippocampus-dependent experiences, one inhibitory avoidance and one novel object location, both of them are functions that require the hippocampus, what do we find? And what we found is that in neither cases there is memory expression. So one inhibitory avoidance that does not capture, is not captured in the maturation by novel object location, and vice versa. Even when we enhance one of these memories, we cannot capture the maturation of the other one. In other words, the maturation goes in domains, the types of learning and experiences we have. And similar data we found if we use artificial stimulation with the DREAD systems. So in conclusion, developmental maturation does not occur by default, it occurs by experience. And the type of experiences encountered during early development, during the critical periods, mature the functional competence of the hippocampus-dependent memory system in an experience-selective manner. In other words, we are the expression of our experiences. There is an important contribution of the type of experiences we encounter during the critical periods. So I want to end. We are, of course, continuing these studies and asking what happens in adulthood now after these early experiences. And I'm going to tell you the story next time when I visit in person. But I want to end by thanking who did this work. And this is the effort of 4 very talented postdocs. Alessio Travaglia, who started together with Reto Bisatz the studies, Benjamin Bessier, who continued in the last set of studies, of which I showed you only one part, and Todd Mowry, who contributed to the electrophysiological studies in collaboration. I thank the funding agencies. Here there are some of our references, and I also want to thank the current members of the lab and say that there are positions open in the lab. So if there is anyone interested, please contact me. Thank you so much for listening, and I'm ready for questions.
Thank you so much for the wonderful talk, as usual, and very different from the last one in the presidential talk in SFN. So I can see that you have different approaches for different problems. So this is a very good one. Thank you so much. So it's open for questions. There is one member from my lab. He said that thank you very much for the talk, that it was very interesting. And have you tried to train rats with a higher intensity to prevent infantile amnesia in rats? Is infantile amnesia an all or nothing phenomenon?
So in our studies, we gave one learning trial, as I showed you, with 0.9 milliamps. Is not very low. It's a medium intensity food shock. The literature before us has shown that if you increase the intensity, or if you give multiple training trials, you still see the rapid forgetting. In fact, the graphs I showed you from Campbell and Campbell comes from, I believe, 30 training trials, many training trials, each of them associated with a food shock. So this rapid forgetting is there if you give multiple trials with multiple food shocks and also increasing the food shock levels.
Thank you. There's another question, and it's asking, what do you think would be the clinical implications of these findings?
Yes, this is a very important question. I'm thinking a lot about the clinical implications because I think we don't have yet deepened into this type of questions because we don't know the biology of it. So the surfacing of these type of mechanisms make me think about neurodevelopmental disorders as a result of problems of critical period mechanisms. This is a hypothesis that needs to be tested. But for example, if the arrest of cognitive functions in neurodevelopmental disorders are also due to the fact that mechanisms of critical period are impaired in the neurodevelopmental disorders, and therefore the functions cannot mature. If that's the case, we're going to have to change our approach to study the critical period mechanisms and neurodevelopmental disorder basis. This is one example. The more we know about what type of biology we have in the brain at this early age and how they change with learning, the more we can create novel hypotheses to be tested in terms of basis for neurodevelopmental disorders.
Thank you. There's another question from Dr. Prado. He said that if very early experiences somehow have effects on adulthood behavior, is this effect context dependent?
We are looking exactly at that. So the new studies that are ongoing is to give the experiences during infancy and then wait until adulthood to see where the memory traces, so to speak. So which type of cells are re-recruited in the expression or manipulation of experiences that the adult animal has. So mechanisms, location, as well as behavioral results. We don't know yet where we are looking at addressing those questions.
And there's a very interesting question also about psychoanalysis. It's Fernando Mendoza. He's saying, thank you very much for the talk. What can you tell us about hypnosis as a method to recover early memories in humans?
Yes. So hypnosis has been, of course, used and is still used in some psychoanalytic areas. It's not really used by all psychoanalysts. The reason why it's not that widely used is that at one point, even by Freud, it was abandoned because it's not easy to understand when it works, what it does, and how we can use it. So for certain things, it's actually very useful, but for the bulk of psychoanalytic approach is not always useful. So getting to the question, how about recovering those memories, is very difficult for now to collect that literature. We will have to go and look at the clinical cases. Clinical cases are described and reference to these individual clinical cases. So it's not collected in a way that neuroscientists like to look at the data. So we will have to collect the data first and then use some sort of scientific approach that we are more used to ask the question. So it's difficult for me to answer that. We will have also to model that in order to understand the mechanisms and formulate testing hypotheses and tests that will address those questions. So clinically, there is some report that says that with hypnosis, we can re-evoke experiences. Whether those are experiences of the critical period or of the infancy, I don't know. I will have to look at the case literature, and I will have to discuss with experts in hypnosis. But it's very intriguing, very intriguing.
Thank you. There's a student that wants to make a question, but he's on. So I'm gonna give him open the audio.
Yes.
Yes, Rafael.
Hello, Dr. Axin. Great to talk. And my question is now that during the development in other brain structure, there are lost neurons. For example, in motor cortex, lost neurons from 20 postnatal day to 30 postnatal day. This switch mechanisms in the protein of synaptic proteins, is possible that these mechanisms occur in some neurons and in others not, and this produce a segregation in these neurons that are maintained by their activity?
It's a very good question. I see your hand, but I don't see you. Nice to talk to you. It's a very good question. So you are getting at the question of what are the mechanisms that are ongoing during these developmental phases that explains all of what's going on. And is there a subset of neurons? Is there a subset of synapses? Are those maintained by the experiences, whereas the other ones perhaps are taken away from the system? We don't know. Those are all good questions that needs to be addressed. It is possible, it's an attractive hypothesis that experience and maturation stabilizes certain circuitry, certain cells, and the others have to go. Because what then remains is our schemas, what I call schemas. And those schemas are experience dependent. It's an attractive hypothesis.
That is all.
Thank you.
Thank you. I'm sorry that I cannot open the video. I don't know why, but there's another question from Alejandro Gonzalez, and he said, this infantile amnesia has been identified or studied in other animals besides murines, besides rodents?
I think there is another rodent species, and then I'm pretty sure there is another rodent species, I forget which one. And then I'm not clear if there are other species. Most of the studies have been done using rodents.
So Dr. Roberto Prado wants you to elaborate on how memory of very early experiences can be maintained to adulthood.
Right. So I see Roberto, hi. We are looking at that. So the experiments now we are looking at with learning in infancy is, as I mentioned, the mechanisms, the region of the brains, the behavior influenced in adulthood from the early experiences. I don't have the final data yet, but we see that changes induced by learning in infancy remains until adulthood. Whether those changes are shifting, right? So if there is a change that induces another one and then another one, we don't know yet. But definitely changes created by learning in infancy last very long, last until adulthood, influence adult behavior. Some of the experiments are already in our paper, for example, with saving. So if we give a saving protocol, which is, for example, an inhibitory avoidance with a very low food shock intensity in adulthood, that doesn't lead to any expression in an inhibitory avoidance days later. So it's a latent trace, basically, and is not expressed. But if the animals had an experience of inhibitory avoidance in infancy, and then we give the saving protocol in adulthood, they show memory. So this suggests that changes occurred in infancy, they last until adulthood and influence saving in adulthood. That's one example, and this has been published, but we are doing many more experiments to understand the mechanisms and what type of changes remain and where.
Thank you. Roberto, you want to say something? Okay, there's one more question. Well, there's so many questions, but Ignacio, someone from a student wants to ask, how is the neural development of the neonate affected by amnesia? So that the neural development affect amnesia?
So I want to make sure I understand the question. Does it mean if we create an amnesia during infancy, what happens to these memories?
When it's in when still there's development in the nervous system. I think that's the question, but maybe he can elaborate a little bit more.
So I'm just trying to give some hint. The infantile amnesia, which means no expression of memory, right, is because the system is not equipped to yet to express the memories long-term, like later, like in the adulthood or later on. But the memories are not gone. They are not forgotten, gone from the system. So they create changes in the system. I think these changes are shaping the way the brain is approaching learning and memory. I call them schemas.
Schemas.
So the difference between the types of mechanism in this critical period versus later are, of course, many. Some I showed, some we keep studying them. One hypothesis that I have is that the schemas requires the stabilization of connectivity. It's an hypothesis that we're going to test. So it's different than the plasticity responses that we see in adulthood.
I see. And do you think that this happened only in the hippocampus? Because there's a lot of plasticity versus other brain regions, let's say the striatum or maybe the medial prefrontal cortex that you say. They are plastic, but not as the hippocampus is.
No, I think other brain regions undergo critical period together with the hippocampus. The hippocampus is just one station that we happen to many in the field happen to study. But of course, the hippocampus is interacting with many other brain regions. I think it's the system that undergoes critical period. And of course, some regions are maturing before others, but that relates to the functions that they are involved in. So I think the crosstalk between hippocampus and prefrontal cortex or other cortical regions or the striatum, they are all implicated in the maturation of this critical period. So looking at other regions would be also important in that sense.
Yeah, and to see what happened, and also maybe the circuits, right?
Exactly. So connectivity, circuitry, I think is one of the possible targets in terms of our hypothesis to see how they mature during this critical period. Once they are mature, then they can function. So I think this is probably one of the interesting questions to look at.
Yeah. Well, Christina, as I say, it has been an honor. Thank you so much for being with us. Hope I can see you not virtual in the future. And it's amazing what you do. And so thank you so much for sharing your information with us. And thank you all for being here. They say that it was a wonderful talk. A lot is saying so.
Thank you all. Thank you all. It has been a privilege. And to the next time in person, I would love to visit Mexico City, but also to welcome you in New York or in Italy. Thank you.
Thank you so much.
Thanks. Bye, everyone.